So my name's Alison Etheridge. My PDF is not loading. Oh, yeah, here we go. And my title is Professor of Probability, which seems really quite appropriate for once, instead of being the butt of all the jokes. So the maths I do is motivated by a desire to understand the way in which different evolutionary forces shape patterns of genetic variation. Okay, so in biological populations. And I've called this random all the way up. So Ian talked about random all the way down, but on the way there he talked about uncertainty of knowledge. And what I'm really interested in is the patterns that are robust to that uncertainty of knowledge. And I suppose Jonathan also told us about order emerging from randomness, order emerging from these very fine-scale events will appear in what I show you now. So what I thought I'd do is just show you some examples. The best way to do maths is by pictures. So this is not exactly what I model, in the sense I don't usually go down to this scale. This is the edge of a bacterial colony. It's a photograph taken by Kevin Foster in the Department of Zoology. This is really nasty bacterium, don't get too close. It's Pseudomonas aeruginosa. And what you can see is this very, very complicated pattern at that boundary. Each of those little rods is an individual bacterium. It's very hard to model things at that scale. And I'm not really interested in things that happen at that scale, because I'm interested in patterns that would emerge over a wider variety of organisms. And so I pull out a bit from there. That's the same sort of colony, it's the same bacterium. But now what Kevin's done is that at the beginning of his experiment, he stained bacteria blue and green, and hopefully in a way that doesn't affect the way they reproduce. And he mixed them together, and he put a drop of this mixture of blue and green bacteria onto the middle of a plate which has been infused with nutrients, and he let it grow. And after a while we see these sectors emerging, and these sectors are what we use as really a proxy for the way that the different bacteria in that colony are related to one another. And if you zoomed in on the edge, you'd see the picture we saw before, but by zooming out we're seeing quite a bit of structure. Now the key things that are leading to this structure are things we try to capture mathematically, and then we do a simulation. And lo and behold, we fortuitously chose the same colours as Kevin, and we get a picture that looks very much the same. We see this classic sectoring, but my bacteria in my simulation here are actually circular. They're nothing like the real thing, and they don't take into account any of that complicated growth at the boundary. And this isn't just something that happens for this particular organism. Here's a picture from Halachek et al. In 2007. This is yeast, and it's the same experiment. Yeast has been stained two different colours. It's been allowed to evolve, and we see these sectors emerging. And I can reproduce a picture that looks more like yeast than like Pseudomonas just by changing a single parameter in my model. So we're capturing something of that local structure in a very simple mathematical model. But we do need some assumptions, and one of the assumptions is that when one of my bacteria reproduces, the offspring are left very close to the parent. So this kind of a model isn't going to work, for example, for a fungus with airbound spores. So I wanted to show this picture because I found it at the weekend. I thought it was cute. But my next grant application, which is currently being written, is trying to model populations like this, where actually the offspring are dispersed a long way from the parent. And then what our simulations show, this will be turned into mathematics in due course, we hope, is a rather complex fractal structure. But it's a structure that you can see is emerging over much larger scales. So what we don't see is sectoring, but what we do see is something interesting. But the most famous example, and this isn't my research, I wish it were, but it's the one that I really wanted to explain to you, is going out to an unimaginably large scale. So I showed you in my sectoring example that a simple mathematical model could capture all these different bacteria. Here's something that captures everything from bacteria right up to human beings. And this is a piece of work which is really due to John Kingman from when he was in Oxford in 1982, which is the year I came up as an undergraduate. So at least I feel almost old enough to talk about it. Now what Kingman was interested in was what happens if I sample not from a bacterium, not from a little Petri dish, a small scale sample, but if I sample from something on the scale of the world from the perspective of my organism. So as humans, suppose I was interested in a copy of a gene that's in me and a copy of a gene sample from somebody else in Europe. Then Kingman's model is the sort of model that we would use. And it's ridiculously simple, and I think it's worth even trying to explain the model that underlies this. So in the bottom left, we have a picture of the model. At the top is a sort of starting population. It's a population of size 10. Think of that as the ancestors of my population. And the population evolves in discrete generations. And in each generation, each individual is just going to choose one parent. And they choose that parent uniformly at random from the previous generation. Okay? So hopefully if I've drawn in enough lines, every circle which represents an individual in the population is connected to exactly one circle in the previous generation, so one layer up. Okay? And you allow this to evolve and you get, even with a population of size 10, a picture that starts to look rather complicated. On the other hand, if I go to the bottom of the picture, what I've done, the bottom of the picture is meant to be the current day population, and I've sampled 4 individuals that have been coloured magenta. Okay? And I've traced back for each of those 4 individuals which parent they chose in the previous generation. Okay? And that's the magenta lines that stretch up the left-hand picture. Now that still looks quite complicated. The lines cross each other. It's not really the information that I want to extract. And so what I've done next is, on the picture in the middle, the magenta picture, I've simplified that by representing the magenta graph on the left as a tree. So what I've encoded in that tree is how many generations I have to trace back before individuals in my sample share common ancestry. And that sharing of common ancestry is what's going to tell me about how closely genetically related they are to one another. So that's why we do it. So you see 2 individuals which I've drawn as if they were in the middle, even though they weren't on the left-hand picture, actually chose a common parent 1 generation back. If I go back a further 2 generations, 3 of the individuals in my sample already have a common ancestor, and 1 more generation and everybody actually has a common ancestor. Okay. Now what Kingman did was he showed how to approximate this for very, very large populations. When M is very, very big, you certainly don't want to draw the picture I've drawn on the left. But instead you can get a picture like the one on the right, which is just keeping track of this relatedness. So the picture on the right is actually a Kingman coalescent for a sample of size 1,000, I think. So what's astonishing about the Kingman coalescent is that actually, if I were to take a population not of size 10, but of size 20, that tree that you see would have the same probability of happening, except for one detail. So I would see a tree of the same shape, but it would be stretched out by a factor of 2, because I've increased my population size by a factor of 2. If I increase my population size by a factor of 100, I expect to have to wait 100 times as many generations to see common ancestry, but the shape of the tree is exactly the same, and the relative lengths of the branches are the same. So this is a very beautiful and rather simple model. But it applies in an outrageous, actually, range of situations. So by changing that single parameter m, which is just changing the height of my tree, I can model E. coli. I would take m to be 25 million. I can model the fruit fly, which every geneticist wants to model. So this is the most common, Drosophila melanogaster. M is 2 million. Humans, well, okay, if you're interested in European populations, about 20,000. If you're interested in African populations, which have much, much more genetic variability than European populations, 50,000. And this poor little chap at the bottom here, the black and yellow spotted salamander, I'm afraid the prognosis is extremely poor because M is something less than 100, and that means there's very, very little genetic variability in that population. But what astonishes me, and it really shouldn't be true, but it is true, is that by varying just this single parameter in our model, we can model this astonishing array of organisms. So that single parameter is capturing natural selection, population growth, spatial structure, and I suppose the role of the research that people like me do is to try to understand how those different effects feed into M. Okay, and that's probably my 8 minutes. I didn't time it.